This is Dr. Irina Conboy. She's a professor of bioengineering at UC Berkeley, where she started in 2005, and she's got quite an impressive resume, which includes being a QB3 core faculty member, which is a consortium which includes UCB, UCSF, and UCSC. She's on the executive committee of UCB and UCSF's graduate program, as well as the research council of the College of Engineering, Faculty 1000, and she is also the editor-in-chief of Rejuvenation Research. I don't really need to introduce her publication record because I know pretty much everyone knows about her work, but she's got quite an impressive group of papers in high-impact journals, as well as she's an inventor on 6 patents and has a productive IP pipeline, which is specific to the field of aging and regenerative medicine. She's received a bunch of awards, including the Silicon Valley Foundation Award for Clinical Translation of Aging Research, Open Philanthropy Award, the Packer Endowment for Aging Research, Raymond and Beverly Sackler TAU Award, the Calico Award, the Bridging the Gap Rogers Award, a SENS Foundation and Life Extension Research Foundation Award, the W.M. Keck Foundation Award, the Glenn Research Award for Research in Biological Mechanisms of Aging, the Ellison's Medical Foundation New Scholar in Aging Award, and the National Research Service Awards. Her laboratory aims to understand the age-imposed and pathological changes in circulatory milieu and their impact on signaling pathways that regenerate tissue maintenance and repair. In the past few years, this direction has expanded to synthetic biology and innovative digital biosensors for diagnostics of age-imposed alterations and for assessing the response to treatments longitudinally in real time. Her work is important because success in this research improves our understanding of homeostatic health and novel approaches to treat diverse pathologies that range from tissue generation to cancer and include novel ways to avoid and diminish fibrosis, inflammation, and metabolic decline. So with that, I'm going to kind of turn it over to Dr. Conboy.
Oh, okay, let's see. Slideshow from start. All right, so we should be in business. Thank you so much for the introduction. I will wear my headphones to make it even better. Do you hear me well?
Very well, yes.
Excellent.
Everyone online hearing okay too?
Excellent. All right, so this is the view from my office. In case you are interested, what I see at Berkeley, I see this courtyard, and I will start with kind of introduction on my general understanding of what controls the process of aging. So some people starting to catch up with this notion, but for a while I have been presenting the slide that our age or biological age, in my opinion, is not really regulated by the rate of damage or how much we are damaged, but rather by the efficiency of repair of that damage. And this is my favorite example when I compare a rat and a squirrel. And so chronologically, these animals could be the same age, 3 years of age. But at 3 years of age, rat will be very old and unhealthy. So at some point, there is exponential increase in deterioration of organs and tissues. But squirrel at the same chronological age will be very healthy animal. Even though they lived in the same entropic environment for the same amount of time. And if you look at the rat and the squirrel, they have similar body size, they have similar metabolism. In fact, squirrel probably has higher metabolism than rat, and their diet is similar, and the predation on them is similar. So none of those conventional theories, in my opinion, explain why would squirrel be so young and healthy, and rat will be so old and unhealthy at the same chronological time. And so the idea is that it is not that the rate of damage increases, but that the ability of repair that damage is better in some mammals as compared to others. And so the Conboy laboratory is focused on this idea. What happens if we improve tissue regeneration and repair in an old person? Would that amount to rejuvenation?
Irina, can I ask you a quick question?
Yes, and feel free to interrupt. It doesn't have to be very formal. It's more like a workshop or a seminar.
Appreciate that. Thank you. In your previous slide, when you're comparing the squirrel and the rat, are we talking about both of the animals living in the wild, or we're comparing rats that are in lab versus squirrels, which are more, not in
I
lab?
don't think it matters, right? I don't think there are many squirrels that live in a lab because people do not usually study them. But the squirrels that are running outside in the wild that we'll see, they live over 20 years. Whether rats, regardless of where they live, they could live in a lab, they could live in the wild. Probably in the wild, they live only like 9 months or so. But in the lab, with a good nutrition and care, they live up to 3 years.
Thank
So
you.
squirrels in the lab maybe would live 30 years. I don't know. So to give you an example, or kind of metaphoric example or comparison of different types of cells that we have, is really nicely represented by this tree stump with the little sprig of new green leaves. So we have this, metaphorically speaking, green leaves, which are stem cells, and those are not embryonic stem cells. They are adult stem cells located in all of the tissues where people try to identify them or study them. So in muscle, we have satellite cells, which are myofiber-associated stem cells. In brain, there are stem cells still even in the adults in hippocampus, in subgranular zone of hippocampus, and so on and so forth. We have stem cells in bone, in the liver, of course, in the bone marrow. And these stem cells are relatively younger than the rest of us because they are typically metabolically inactive, so they don't damage themselves very much. They have capacity to activate telomerase when they divide, and so their progeny has normal telomeres. And they also have very high fidelity DNA repair. So this is our hope, I think, for rejuvenation. And then most of us are differentiated cells. They accumulate damage and they do not have telomerase, so they do not maintain their telomeres. But at the same time, they are not losing telomeres because they are not dividing. And we cannot lose telomeres without replication or cell cycle. And then the worst of those damaged cells we call senescent cells, where DNA and other damage is high, telomerase is inactive. There is also SASP. So they secrete proteins that are pro-inflammatory and are resistant to cell death or apoptosis. But what was shown very definitively by our laboratory and then repeated throughout the world is that other heterochronic parabiosis, which is shown here by this Halloween costume, or more advanced approach of heterochronic apheresis, which was indeed supported by Stem Cell Research Foundation in our lab, both of these approaches can reset age of tissues in an old animal back to youth. So regardless of what happened intrinsically, it seems that there is a lot of plasticity in our regenerating system to turn from an old system to young system. And vice versa, if you start with a young animal and expose it to old blood, young animal becomes prematurely aged. Again, confirming that it's not simply a progression of time that controls the process of aging. And then in the more recent work that we are doing in the Conboy laboratory, we no longer use exchange between one mouse and another mouse, but we exchange an animal with the designer solution. So here is the next generation small animal blood exchange device, which is programmable. And as you can see, there is just a tube with a particular solution, and we can exchange an animal with that solution to a specific percentage of exchange at specific rate of exchange. And how we do it is published in Nature Protocols in case you are interested. So there was a lot of discussion whether young blood is a medicine or we need to remove old blood. And in vitro experiments as well as in vivo outcomes to us suggested that when two animals share blood, either through suturing them together or through blood exchange, it is not the young blood that rejuvenates the old mouse, but it is removal of the old blood by the young partner that is rejuvenative. And as you can see, this is, again, metaphor for glass half full, half empty, when there is parabiosis or blood exchange, you have a young mouse that is half full with old blood, half full with young blood. You have an old mouse that is half full with old blood, half full with young blood. So absolutely, there is no reason to believe in this cartoon, which is, by the way, is press, positive press by The Economist about one of our stories, which shows that there is an army of old people who tried to sneak in blood donation center and steal this young person's blood. So though intuitively it seems that the most superficial interpretation of these experiments is that young blood is a medicine, it could be either or. And with respect to either or, this is the work that was published in Rejuvenation Research last year, by the way, from the laboratory of Irina Conboy, who is professor in Kiev, Ukraine. And most of this work was done in her refugee status. So we were happy to for them to contribute in such a meaningful way. But in itself, this is an amazing work that I have been asked at conferences many, many times. So what happens if you disconnect parabiotic animals? Do old mice live longer? And what you can see here is that it's compared to the isochronic old to old parabiosis. Old to young parabiosis do not live longer. This is the dashed red line versus solid red line. And in contrast, as compared to isochronic young to young parabiosis, young mice that shared blood with old mice for a few months now experience shortened lifespan. So that would suggest that young blood really does not overcome deleterious effects of aging, but old blood has progesteronic effects even on young animals with all of their undamaged DNA, long telomeres, whatever else you can think of. So I would like to plug in my work as editor-in-chief of Rejuvenation Research and ask collectively of SENS Research Foundation to perhaps send us your vision on what is the process of aging and how SENS Research Foundation plans to tackle it. Please send us a description, whatever you would like to put forward. What are your goals and objectives? We are looking to such publications. And also for everybody attending this workshop, please think of a review paper that you might submit. We really would appreciate it. We try to restructure the journal and make it of a higher impact, meaning that we don't really care what the impact of a journal is. We are on the same page as Randy Shackman and other Nobel Prize laureates, but we care about the impact of individual papers. Please send us interesting papers that are innovative and good research or provide innovative perspective on a field. So to moving forward, even though we speculated or hypothesized that young blood is not needed, we decided to actually test this hypothesis experimentally, and now we had this approach where we could have young or old animal, and instead of exchanging them with young or old blood, we can exchange it simply with saline and 5% albumin. I have to mention here that albumin is not anything that we are adding. We are simply replenishing the albumin that is taken out from the animal. So when we are taking out 50% of animal blood, we discard all of the liquid fraction, which is plasma, and it contains the major protein called albumin. And without albumin, animal will be very unhealthy. So we are simply replenishing this albumin with the commercially purchased powder albumin. And we return blood cells from the same animal isochronically, such as that at the end of the experiment, young mouse plasma is diluted by 50% and it gets young blood cells back. And old mouse plasma is diluted by 50% and it gets old blood cells back. And this paper, which is, by the way, published as a priority research, and it is highly cited already, even though we published it during pandemic, so it has been barely 2 years, showed that old plasma dilution rejuvenates stronger than all other current approaches. It makes old tissue statistically the same as young. So without any young blood, without GDF11, without TIMP2, without any proteins, simply diluting old blood plasma of old animal by 50% is rejuvenative. And we published follow-up paper that focused specifically on cognition and neuroinflammation, showing that the same process improves cognition and attenuates neuroinflammation. And I will show you some results from mice and then some results from people. So in mice, I'm not going to show you all of the results back from 2020, but just to summarize, there is rejuvenation of muscle with less fibrosis and less adiposity in liver, and then more hippocampal neurogenesis, and then reduction of neuroinflammation and brain senescence. So many attributes that were studied by different laboratories under hypotheses of different biomarker or biological determinants, they all return to youth and health when we simply dilute all blood plasma by 50%. So to show you some of the more recent results, which were published in 2021, this is one of the behavioral tests that we do, where mice are asked to identify a novel texture or novel object in their cage, in their environment. And they do it after we performed either what we call neutral blood exchange that I described. Neutral blood exchange stands for neutral age. So we simply dilute plasma by 50% with saline and albumin. And our control experiments are isochronic blood exchange, young to young and old to old, which simply control for the procedure. And then we do this schematic that is shown, we perform experiments schematically shown here on top. So we perform exchanges and then we test behavior. And what happens is that typically young animals are very curious and they know which one is the new texture and new object right away, and they spend more time with them. And old animals either do not remember which one is young, which one is old, or they don't care. Kind of like young and old people in a way. And then after single procedure of neutral blood exchange, now old animals are statistically the same as young in this cognitive test. And the same procedure, if you look at the SA beta gal positive blue senescent cells in the brain, reduces the numbers of senescent cells to the same degree as non-senolytic ABT263. So there is much fewer of these blue dots in brain parenchyma, which signifies senescent cells. And then we also looked at neuroinflammation, which we measured by the presence of CD68 expressing or CD68 high, CD68 microglia cells in the brain. And the schematic shows where, in which regions of the brain we found microglia that is activated. And then we quantified all of the serial images of the brain to the same conclusion that neutral blood exchange in old animal makes them statistically the same as young animal, whereas there is increase of neuroinflammation with age in control animals, as one would expect. With respect to the hippocampal neurogenesis, which I mentioned, we measure it by the presence of dividing, shown here, Ki67 is the proliferation marker, dividing neural precursor cells in this particular area, subgranular region of denta gyrus of the hippocampus, which looks like a V. In the old animals, and this is isochronic old, there are very few of these cells, but then after neutral blood exchange, you see increase in numbers, and this is antibody control. And what you can see is that in the control young animals, there are more than 10-fold higher levels of the hippocampal neurogenesis as compared to control old animal. And then after single procedure of neutral blood exchange, the levels become similar and once again, non-statistically different. So it is really truly rejuvenation. Another point is that young animals whose plasma is diluted, on average, do not become worse off, but there is significant variation now. And it is also true, although to the lesser extent for muscle and liver, which is in older data. So overall, dilution of the young blood factors is not very deleterious because young animals can just express higher levels of those, making more mRNA and protein in a relatively short time. But dilution of the aged blood factors is hugely rejuvenative. And the same cannot be said about senolytic. So in senolytic approach, there is no rejuvenation of hippocampal neurogenesis. So even though there are senescent cells in the brain, and even though they could be removed with senolytic ABT263, we don't know what happens, but there is no improvement in neurogenesis or neuroinflammation. So when we discussed these results with Judy Campisi, she told us that ABT doesn't cross blood-brain barrier, but still we see removal of senescent cells in the brain because probably ABT reduces senescent cells in the periphery and thus reduces SASP, proteins which go to the brain and induce senescence and trans, which I will discuss in the next couple of slides. And by the way, dilution of blood plasma does not cross blood-brain barrier either. We simply dilute peripheral blood, right? But somehow that process reduces senescence in the periphery, reduces senescence in the brain, and rejuvenates neurogenesis and diminishes neuroinflammation. So we still are working on understanding these processes, particularly in collaboration with my former postdoc, who is now a rising star in bioengineering, Professor Kiana Aran. She's associate professor at Keck Graduate Institute in Pasadena. And so the work we published with her laboratories focuses on the blood-brain barrier organ chip. So here we have the brain part and we have the blood part. Blood part is particularly physiologically important because there could be different shear stress on blood components, and we can look at the effects on the cells of the brain, such as neurons and astrocytes, and also on a barrier to see if permeability of the barrier or integrity of BBB becomes worse off or better. So this, I think, is our next step in studying mechanistically what happens with neutral blood exchange and senolytics when we are talking about effects of blood on the brain.
Can I ask a quick question regarding?
Yes, of course.
Regarding the systemic effect of senolytic drugs in reducing inflammation?
Of course, yeah.
Right? Do you guys plan on looking for infiltration of CD8 T cells or NK cells in the brain? Because in older brains, these immune cells tend to migrate and can cause damage, and this perhaps could be an explanation or another way to confirm that it is actually reduction of SAS, which is
yes, very good points, right? So it probably would be cost prohibitive and super difficult to do in vivo in mice, but with the blood-brain barrier that we are actually currently humanizing, all of that would be very good to study because, for example, the part of the chip, this is the organ chip. So the part of the organ on the chip which has blood in this chamber, we could introduce the whole blood, we could introduce blood cell subsets, and we could look at their permeability to the brain.
That is a good experiment.
Right.
One could also do histology though in the brain and then stain for these markers. Even H&E stain would be able to reveal some of these.
We could, right? But as far as I understand, if I understand correctly, it really the blood-brain barrier should disintegrate significantly, and the migration of T cells and B cells into the brain is the indication of a disease, for example, multiple sclerosis or ALS. So in the physiologic old animals that are relatively healthy or people who are relatively healthy, I would not expect to find many leukocytes outside of the resident microglia in the brain.
Well, no, there are actually, Irina, there are actually a number of reports on this already with T cells in particular infiltrating the aging brain and doing a lot of damage, including being apparently responsible for hippocampal damage and the decline of hippocampal neurogenesis. I think that's probably the background for Amit's question. It would be really interesting to know.
Is it even without, like, multiple sclerosis? Just
or
Alzheimer's, just like normal.
Quote unquote normal aging mice. Yeah, 100%.
Normal aging mice. I see. So yes, so thank you guys. Very, very, very important information because we plan to look at PBMCs in BBB organ chips that we have, and we are optimizing those. But certainly our connection from organ chip is always to the in vivo system, and it's important for us to know that perhaps there are T cells migrating to brain even outside of normal pathophysiology of neurodegeneration or diseases.
There is some more limited data, even in humans, that are not diagnosed with any specific neurodegenerative disease. Although, of course, like, it's much more flamboyant when you look at
Yes, exactly.
people with,
Yeah, good. So that will be something to, if you are interested in that, certainly to pursue further, or if not, just thank you for providing this information.
Absolutely. And another comment regarding that, I mean, neuroinflammation is becoming really, really, like big now, and we're starting to realize how complicated this is. There is a subpopulation of NK cells that reside in brain, in Dante's gyrus, and they tend to also be affected by neuroinflammation and aging. And that would be another like reason to maybe look at the histology of the brain, and see if this partial blood exchange or just supplementation of albumin would improve that, because that will go
Yes, absolutely. All of that, right? So providing more holistic window into what's going on with the neutral blood exchange and brain neuroinflammation instead of just looking at microglia activation. Absolutely, I agree. So then shifting gears, and this is a very nice segue. Now I'm going to tell you what happens to young animals that are exchanged with old blood. That is the kind of the other side of the same coin, that if dilution of age-elevated proteins in plasma is rejuvenative, then when we introduce old blood into young animal, we expect them to be prematurely aged, and that's indeed what we observed and published recently. So this work was done mostly by Oki Jion, who did brilliant postdoc with Jennifer Elisef and then Judy Campisi. She published seminal papers in both laboratories, and she did the lion's share of the work on this paper. And the work was done over pandemic while she was already running her laboratory in Korea. So even more glory to her that she decided to see it through. And so the first piece of data that I'm going to show you is that old blood serum makes young cells and young mice senescent. So in these experiments, we have serum from either mice or people, and we apply them to cells which are non-senescent, ordinary cells, and then we measure markers of senescence, basically calling it senescence transfer. And the cells have not been irradiated or UV irradiated or overgrown, so there is no other stimuli for them to become senescent, except that they are cultured with old serum, and the cells are young. So, so then we measure P16, P21, IL-6, MMP3, and lamin B1, and by all of these mechanisms, old serum makes young cells senescent. And then the same happens when we look at human cells, and this is just SA beta gal. Human cells will become SA beta gal positive just when they're cultured with old plasma, even though they are young cells. And then we can look at proliferation, that's also consistent, and this is mouse cells, SA beta gal, and this is some images. But the take-home message is that, and I put this in exclamation point, that old serum dominates over young when they are mixed. And I put exclamation point because, first of all, it's super important mechanistically, but also because Mike Conboy and myself have seen it, have observed it back in early 2000s when we were working on our Nature manuscript of heterochronic parabiosis. We did all of these experiments before even doing parabiosis. And now I'm happy that his laboratory reproduced them. So basically the same markers, P16, P21, IL-6, MMP, lamin B, and then EDO or proliferation. And so this is young, this is old, young plus old. It dominates. This completely dominates, completely dominates, completely dominates, completely, and completely dominates. So even though the young serum is also present, factors, and we think those are proteins in the old serum, dominantly make young cells senescent by all measured parameters, and it is not just one parameter, it is few of them. That again puts a big question mark on all of these approaches where old animal or old person is injected with something young. How can that make them young when there will be many, many dominant inhibitors of the positive effects? And when all of their cells and tissues already are damaged, where here we look at totally young cells in preferred culture conditions, they just simply have exposure to old serum alone or a mixture of old serum with young serum. So that is something to think about. And then another point is that when we treat mice with senolytics, we see that yes, senolytics are effective and we reduce SASP proteins wherever we measure them. So this is dashed line is how much they were, and these are the new levels. So senolytics really work to reduce SASP proteins. But when we look at the effect on muscle regeneration, we see no positive effect. So this is the newly regenerated muscle tissue after injury, and this kind of nice pink looking is the one that was not injured. And we do injury by injections of cardiotoxin, which is a little droplet of a snake venom. And so the white areas are fibrosis, the dense blue areas are inflammation, nuclei of infiltrating cells. These red clusters are newly formed muscle fibers. And when we compare vehicle versus senolytic, there is absolutely no improvement of old muscle regeneration. Whereas with neutral blood exchange, there is a huge improvement. So we published it, and I do not show it here again.
I have a question.
Yes.
Muscle, the heart muscle regeneration, is it something that actually happens efficiently in young mice at all?
Yes, it happens in young mice very actively, and I apologize for presenting a short talk today. I could have shown what happens in young mice and also what happens to young mice exchanged with old blood and all of these other things, but I decided just to run through various approaches under one major umbrella of what controls process of aging. So young mice regenerate very well, and at this time point, which is 5 days after injury, this whole area would be full with these little newly formed muscle fibers. They would look exactly like those guys, but very tiny and with central nuclei, and there will be no fibrosis or inflammation. So yes,
Irina,
go ahead.
sorry, the related point I wanted to ask you about, and I had mentioned this at one of the online conferences that we had both attended a little while ago. There are now a couple of other groups that have reported that senolytics do in fact improve muscle regeneration in old mice, and interestingly, the mechanism seems to be in one case via ablating the macrophages that are inside the cells rather than actual senolytic cells, senescent cells. Have you looked at those at all? And do you have any idea why you get contrasting results?
So we published, I think, maybe 10 papers or more on how to make old satellite cells to become younger and regenerate muscle better. So there are numerous approaches: heterochronic parabiosis, heterochronic blood exchange, neutral blood exchange, inhibition of TGF-beta, inhibition of osteopontin. So there are many, many ways by which old muscle can regenerate better. But ablation of senescent cells with senolytics, which we did here, and collaborative work with Judy and Oki, is not one of them. So simply adding senolytics, this is what was done, and it was done by experts in the field, is not sufficient to make old muscle regenerate better. The same is true about the brain, right?
I'm just saying a couple other groups are reporting contrasting findings, and I'm wondering if you have any ideas as to why there might be different labs getting different results on that front.
I think that maybe specifics of experimental approaches, what was done, how it was controlled, how it was measured, right? So so the only thing we can say is that if you do things like that, which I'm showing you here and it's published, you guys can scrutinize it. This is the results you will have. If you do things differently, then I'm not arguing that the results could be different.
Yeah, Michael, I agree. I think we will have to carefully examine the drugs they use as senolytics, the duration of the treatment, how soon the treatment was starting, in order to show an improvement in muscle function. I think a lot of those things will have. We should look at those papers very carefully and see if there are experimental discrepancies between
Yeah,
them.
and also muscle function could improve, right? For example, if muscle regeneration is not better after cardiotoxin injury, if you do not injure muscle with cardiotoxin, muscle function can improve because there are better neuromuscular junctions, muscle innervation or vascularization. So all we are saying is that if you look at the regeneration, it's not better. And so that then brings me to the last bit of the discussion. Once again, because the papers are published, I kind of rush through them, but feel free to ask me more questions by email or anything that needs clarification. And this is what we published most recently in Neuroscience, applying this neutral blood exchange to people in a clinical study in collaboration with Dobry Kiprov, who is performing all of this therapeutic plasmapheresis in people, and Joel Kramer, who gave us, Dobry gave us samples of people who underwent this procedure under IRB approved study, and Joel Kramer, who is the director of UCSF Brain Aging Center, gave us many samples from young and old individuals who do or do not have neurodegenerative disorder or mild cognitive impairment under the MTA agreement. And the work was done by Kim Dahwan Kim, we call him, and Connor Llewellyn, who, so Dahwan is a postdoc in the lab, and Connor was a rotational student. So he did this work during his rotational lab. So clearly he is brilliant. All of these people are outstanding collaborators. So what was done there in this paper is that we looked at the rounds of plasmapheresis in individuals who were generally late middle age or old people. And we compared their proteome and also the presence of specific markers of PBMCs in their blood and other metrics of aging, other biomarkers of aging, as time progressed and they underwent several rounds of the procedure. So here, each colored line is one individual. As you see, for some of them, we did not have the first couple of rounds, but we had later ones. Others we had the most, and others underwent only a couple of rounds. So each dot is a round of procedure where 70% of their blood is removed, and then blood cells are returned back to them in physiologic solution, which is saline and albumin. So mechanistically or methodologically, it's very similar process as I discussed in neutral blood exchange. But it is FDA approved and is routinely in clinic, but it is used usually to treat serious diseases, and Dobry decided to apply this procedure to relatively healthy older adults to see if they also will be improved and rejuvenated, as far as I can understand. So as I mentioned, we studied several biomarkers. One of them is DNA damage, 8-hydroxy-DG. And by this assay, most of the participants had reduced DNA damage of their blood cells, and typically it just went down significantly after just the first procedure, the second, and so forth, and it continues to go down, so that's a good sign. Senescence of PBMCs, I hope you guys know what PBMCs is, right? Peripheral blood mononucleate cells. Typically, those are leukocytes, such as macrophages, mostly macrophages, and some B cells and T cells. So DNA damage in senescence declined. Numbers of T cells increased when we looked at the T cell receptor CD3 or the co-stimulatory molecules CD4 or CD8. Those are two different types of T cells. And there were more B cells when we looked at different markers, more natural killer cells, and more importantly, the ratio of lymphoid to myeloid cells increased, and the ratio of lymphoid to inflammatory cells increased, or natural killer to inflammatory cell increased. So all of these are good indicators that the overall immunity improved in people who underwent several rounds of plasmapheresis. Again, this is subject 1, 2, 3, 4, 5, 8. The work was done with 8 subjects. It's a pilot study. And each dot is a different round of the same procedure. And then what is important here is that we also performed blood proteomics, and we compared rounds of plasmapheresis with what happens to old people as compared to young people. So Dahwan made this nice colored figure where you can see how aging is pretty much diluted by the rounds of plasmapheresis. So we start with the load of these orange and red squares, which means that particular protein, that are listed on the right, of a particular signaling pathways, which are listed on the right, on the left, is elevated. And this is 1, 2, 3, 4, 5 different old people. These are also old people before they started plasmapheresis around 0. These are the same old people after 1 procedure, after 2 procedures, and some of them, very few, did more than 2 procedures, 3, 4, and 5. And these are young people, 1, 2, 3, 4, 5, 6. And what you can see is that gradually for everybody, and typically by around 2 or 3, the age-specific pattern of blood proteome becomes closer to that of the young person. And that is very important because all of the signal transduction networks do not work just in bone marrow or blood cells. They broadly coordinate health and repair of all of the tissues throughout the body. So systemic proteome here is indicative of overall health of a person. Not, proven to be so, but indicative. And then Dahwan identified toll-like receptor 4 as the only crosstalk node between all of the signal transduction pathways that are influenced by therapeutic plasmapheresis. And not surprisingly, when we look not just at 8 people, but 350 people who are young or old, TLR4 is age elevated. So it is super important that the levels of TLR4 are diminished stably after many, many weeks of procedure by therapeutic plasmapheresis. And I mentioned that we also got some blood samples from UCSF brain aging clinic. And so when we plugged it into the multidimensional UMAP plots, we identified that these are the people who are old and have neurological disease. They stand apart from the rest of the groups and their clustering of the proteome. But then when we look at this group, which is old, or another group that is old, it is therapeutic plasmapheresis around 0. So it's old people who came to Dobre Clinic and donated blood before any procedures. These are very cluster close to each other. But then after plasmapheresis, now the last round of procedure in old people makes them close to middle age group. And kind of everything shifts here, not as young, but still at least middle-aged.
Irina, do we know
Yes.
how after the last procedure the sample was collected? I'm actually trying to get to how long the effect lasts after plasma.
Yeah, so we did really good job in this paper by publishing figures in supplementary information, exactly how many rounds, what is the interval between each round, and from that you can figure out how long the procedure effects last. But I would say that the last at least 1 month, because that was the interval between each round. So this was before first round, this was 1 month after that, this was one month after that, and so forth.
Thank you.
Mm-hmm. So moving on, just to the last bit, I think this is my last slide. So then we decided to measure biological age of individuals. And here I would like to tell you the difference between predictions and measurements. So here is the cartoon of a typical county fair, right? And there is a guy who can predict your weight. And we can predict weight, right? We can look at how people, what their body size and shape is. For example, I can predict that the weight of this individual is larger than the weight of this individual. We can also predict weight based on what we eat. I can certainly make predictions. But predictions as population statistics method will never be as accurate as measurements. So instead of guessing the weight, if somebody simply stands on the scales, then I don't even need to look at them. It could be blinded experiment. I could just look at the scale and tell how much they weigh. So our goal in our laboratory for many, many reasons is to move away from predictions to measurements. And so what we decided to use, and it's also published in the same paper, is biological noise, or what is the standard deviation of particular proteins. Can we identify the detectors of that noise? For example, this protein, UPAR, is a good detector of biological noise because the biological noise is high from old to disease, and it goes down as compared to before plasmapheresis to after plasmapheresis, and it goes up when we look young to old transition, and middle age is someplace in between. So simply by estimating, by looking at hundreds of proteins in hundreds of individuals and identifying those that will detect biological noise, allowed us to identify those detectors. And then now you can see that when we simply measure standard deviation values of the biological noise, we see that for each one of these detectors, biological noise increases from young to old. This is not a prediction. We are simply measuring the noise of these proteins. And from that, we can move forward to actually measuring biological age. Here is relative years. The relative years are measured just based on the noise or standard deviations of 10 detectors. And we see that it is low in young individuals, which is good. And then in the middle age, it becomes higher, and moreover, it is not extremely identical. So some have higher level of biological noise, others have lower level, and that makes me very happy because that's what I expect from 90 or so years of modern biomedicine. And it goes further with age. Now we have, instead of tight grouping, we have kind of a fanning effect. And then if people have mild cognitive impairment, so this is from UCSF clinic, now even though chronologically they are here, but biologically they are way up, this line, by the way, is identical to this one. It looks lower because there are more dots on the screen. And interestingly, in therapeutic plasmapheresis, it's almost a mirror image. So pre versus post, pre is above the line and post is below the line. And so by the measurement of what we consider biomarker, which is biological noise of the proteins, all of the individuals became significantly younger. This is, again, not prediction, it's a direct measurement. And you can kind of see it as a raw data here. So this is 40 to 50 years old before plasmapheresis, and they have high level of noise of the specific proteins. This is 60 years old, 70 years old. And then after plasmapheresis, there is some effect on middle-aged people. Biological noise is decreased, but more effect on old people. Their biological noise of the proteome now becomes very, very small. So this is our approach. I don't have time, or we discuss only published data, but we continue to work on this, taking it forward as an alternative to predictions. I think measurements should be better than predictions whenever we do that. And all right. And so this is just my conclusions. I don't have to read through them again. I told you about rejuvenating effect of large dilution of systemic milieu. I told you that old blood dominates over young in vitro, and that blood dilution does not act simply by attenuating SASP, because there are huge difference between senolytic and blood dilution. What we published, and I did not really show you, is that senescence can be induced in 2 weeks in young mice with all of their young blood cells and tissues simply by single blood exchange with old mice. And then I ended on parameters of chronological age, that those are no determinants of biological age. We have chronological age and we have something else. And this is the wonderful lab, and many thanks to our funders, and thank you so much for listening to me. Sorry, there was some technical issues to begin with.
No, that was wonderful. Thank you, Irina.
Yeah.
Does anyone have any questions here or online?
I have a question also. Thank you for a very interesting talk. So I was wondering if those factors that you listed in the end that were predictive of the biological age, are they also predictive of survival?
Survival. Yes, of course. So basically, what I want to mention is that survival depends on biological age. It's almost like, if somebody plots biological age versus survival outside of crime and accident, they will form one line, as far as I can tell.
My question is, if you do this blood exchange or rather plasmapheresis, what would happen to those factors? They will all go down?
Yes. So and that is again published, and I apologize for not introducing this data. I should have. So what happens is that when we have one huge dilution of, let's say, old protein in old plasma, that is inhibitor. Usually such proteins have positive feedback for their own expression. So the same protein will bind to a receptor and then induce its own mRNA and more of that protein. So by diluting it, we break the positive feedback. And then that produces stably diminished level of that protein for a few weeks at least. So that's what we identified in the 2020 paper. All very important questions.
Yeah. What if you delete also the factors that are good for the body?
Well, in an old animal, there is not many of those factors, right? Because they have age-elevated proteins that inhibit their expression. For example, TGF-beta that is elevated with age then changes productive signaling through MAP kinase pathway by many positive factors, LIF1, for example, leptin, changes those signaling to its own overproduction of TGF-beta, which is called non-canonical MAP kinase. So that's why with age, you have less and less of oxytocin, positive juvenile hormones, and so forth. It's not only that their regulation is diminished, but their inhibitors are up. So what we are doing, we are actually inducing their expression. That's also published. That once we dilute age-elevated proteins in blood serum, they cannot inhibit their targets. And as the result, now we have rejuvenated levels of the young factors, because they did not disappear from our genome, right? They're still encoded, just their expression was inhibited and now it's de-repressed. So in theory, we don't need really introduce anything ectopically young, in theory, because the useful gene expression is restored by removing once and at the very large levels, circulatory inhibitors of those factors.
So what would be the mechanism of action? Like, how would this be working mechanistically? Is it that you're removing these inhibitory factors and then the immune system can then remove senescence burden? Is that what's happening?
So yes, we are not removing inhibitory factors. We are let's say we remove them, we dilute like a lot of them. And so what happens is that these factors are signaling molecules for signal transduction networks. Once we remove them, we perturb their positive feedback for making more of themselves, but we also prevent them, because we remove them, from inhibiting all of those good things that decline with aging. Those good things do not simply become less for no reason. They become less because TGF-beta, for example, is increased and inhibits productive conventional MAP kinase signaling by numerous other proteins, and inhibition of that signaling has deleterious consequences for gene expression. So pretty much you can look at it as a reset, like you reboot your computer.
Right. So that would explain that there is no increased senescence burden over time when you do that. But you're seeing a reduction in the senescence burden, which means there is an active removal of senescence cells. And to that I was asking if
I don't know. Yeah. It might be that well, what we look at SA beta gal, right, and P16. For P16, I can tell you for sure, P16 is not a marker of just senescent cells. It's just CDK inhibitor, right? So what we see is that now there are less cells which are inhibited in their cell cycle progression, which makes sense, right? So then regeneration is better. As a beta gal, it is a little bit more surprising, but I have heard at the conference on senescence that as a beta gal can go away as well. For example, you have overgrown fibroblasts, which are as a beta gal positive, and you split them and they become as a beta gal negative. So I don't think that the only way to deal with senescence is to ablate them. Neutral blood exchange or TPE could work in a way as synomorphic, not senolytic.
I see. Okay.
Mm-hmm.
So if senescent cells do not play a role in producing these harmful factors, where do they come from?
Say it again? I did not.
If senescent cells do not play a role in producing these harmful factors, where these harmful factors come from?
Well, senescent cells produce SASP, but I don't know how many senescent cells we have in vivo. And how much.
But if you remove them, you said if you remove them, that does not affect the regenerative effective deletion. So that means those factors must come from other cells.
Oh yeah, absolutely. So basically, let's look at it as a continuum, right? So, and that's why I introduced this first slide, is that you have differentiated cells and they experience damage. And when they experience damage, they will produce all of those molecules such as transforming growth factor beta, right? Absolutely, even if they are not officially senescent, they are just terminally differentiated. And then it just differentiated niche. And the longer differentiated niche persists and the more it is damaged, the more of those inhibitors will be produced. And then some of them will become totally horrible and senescent, but not all of them. It doesn't have to be that way.
Okay, thanks. And how about the reversibility? When you do a plasma dilution and you have this beneficial effect probably after 1 week. So how that will last and when these damaging factors will populate again the plasma?
Yeah, so that requires longer study. Again, if you would like to collaborate on that, that would be great. Dobry Kiprov is doing now big sponsored clinical trial where it is a study with mock plasmapheresis versus real plasmapheresis in 40 people. I am not part of it, so I cannot tell you what their endpoints are or what they plan to do. But the ultimate goal is certainly very, very noble and important, is to see if procedure could be used on relatively healthy older adults. From our perspective, we know that the effect lasts, as I mentioned, at least 1 month in people. But the word of caution is that we need to understand side effects much, much more. And I receive like so many emails from individuals almost every day, not every day, every week, who would like to do this procedure to become younger based on our work. And I keep telling them our work does not tell that it is safe. We studied very limited parameters. For example, heparin is always used in the procedure. So there will be effect on blood clotting, right? Blood clotting factors levels change by this procedure. So they might be elevated at some point after procedure, and that would cause a risk of strokes, both, the thinning blood and blood clots. There are many other things which in my opinion should be studied at the level of the whole organism, like MRI, functional MRI. From my perspective, I don't think that anybody will become stably younger and healthier just by taking 70% of their blood, shedding the cells in this process, returning the damaged cells back in saline and albumin. From my perspective as a scientist, it's just the scientific knowledge. It's the paradigm shift. We don't need young blood to become younger, and many parameters become better and younger and statistically the same as in a young animal simply by diluting plasma proteome. That's it. So that's my take-home message, and from there we take it into understanding the mechanism so we can do it pharmacologically.
I had a quick question. This is more just like general understanding, but like when you're doing the plasma dilution, you're not diluting the number, the blood cells at all, just the plasma, so the number of cells stays the same.
Well, they're kind of the same, but not really the same, because, for example, red blood cells, which is most of our blood cells, will be sheared and damaged simply by your blood being taken out from your veins, from your body, going through some plastic tubing, and either filtered through a mesh or spun in a centrifuge, depending on what machine is used. And then whatever is left over, whatever there's platelets, they probably will clot or degranulate. There are many other cells that will not be the same, but the age of the cell will be the same, but it will be the gamish of cells which went through the machine and is returned to you.
So there are less, so the concentration of cells in the blood is lower than when you return it into the body, or am I not understanding that?
I don't know. I think I have to guess, right? So, so I'm not a clinician. But, and again, I would like to say that this procedure is FDA approved only for serious diseases. For example, somebody is rejecting their kidney or liver. The procedure is performed to remove the antibodies. Or somebody has multiple sclerosis. The procedure is used to remove autoreactive antibodies. So it is not used on relatively healthy people. And in the FDA approved process, it is used, responsibly. Otherwise, the patient will be worse. Now, when we're talking about cells certainly would prefer not to go through tubing, filtering, and centrifuging by a machine. How many cells are returned? Roughly the same. How many of them are in the same health? I don't know. Yeah. So all of that, kind of to me tells me that I basically, answer to all of the emails from putative patients is that have discussion with your primary care physician. This is not approved. Wait until there's clinical trial and so forth. Yeah.